Hello, everyone, and welcome to this Genetic Engineering and Biotechnology News webinar. It's entitled, Harnessing the Power of High-Resolution Melting Genetic Analysis, from Bench to the Clinic. Increasing numbers of researchers are turning to high-resolution melting to carry out studies involving the genotyping of SNPs and the scanning of sequences and DNA samples. The method itself is based on comparisons of the dissociation profiles of PCR products, hence the term melting. A number of investigators cite the development of improved double-stranded DNA binding dyes and advances in PCR tools and technology for the growing adoption of the technique, also known as HRM. The result they point out is a method that serves as a cost-effective and faster alternative to many traditional post-PCR genotyping approaches. Today's webinar was designed to provide you with an overview of the power of HRM and to demonstrate its capabilities over a range of life science applications. We're delighted to have with us a panel of scientists who have become quite expert in the utilization of HRM technology. Let's meet them. Dr. Adam McCoy is Senior Scientist in the Gene Expression Division at Bio-Rad Laboratories. Adam will talk about the breadth of HRM applications all the way from gene scanning to gene identification. Dr. Jonas Winchell serves as the Chief of the Response and Surveillance Laboratory of the International Emerging Infections Program at the U.S. Centers for Disease Control and Prevention. Jonas will also discuss HRM applications and show how the technique's flexibility allows researchers to quickly identify genetic differences, including antimicrobial resistance, genotypes, speciation, and SNPs. Dr. Kim de Lanier, a scientist at the Center for Medical Genetics at the University of Ghent in Belgium, will look at the application of HRM in a diagnostic setting, specifically in BRCA1 and 2 mutation scanning. I'm John Sterling, Editor-in-Chief of GEN, and I'm going to serve as the moderator. Please feel free to enlarge the slide images or download the complete presentation. At any time during the webinar, you can send in a question for our panelists. Type your question into the Ask a Question box on the lower left of your console and then hit Submit. The panel will try to answer as many as possible during the question and answer segment that takes place after all the presentations have been made. Okay, so if everybody's comfortable and sitting in front of your computers, I think we're set to begin. Adam McCoy from Bio-Rad will be the first speaker. Adam, the floor is yours.
Thank you for the introduction, John, and thanks to all of you who are taking the time to join us in this webinar. My job today is really to demystify the HRM analysis a little bit and help you focus on the key elements that are important when trying to improve your current analysis or when designing a new assay. Next slide. There are two broad areas of genetic analysis that are really quite different in their goals. Gene scanning, where you're looking to find variation. You may have no idea what level of variation might exist among samples or which bases might be different, and you're really working in discovery mode, trying to figure out which samples are interesting enough to look at more closely. And HRM can be helpful with this because it's fast and low cost. Or you might already know exactly which SNPs you're interested in, and maybe you want to know if this particular patient sample has a copy of one particular allele. These are very different types of questions, and while it demonstrates the power of HRM that it can be a useful tool in both of these areas, it also presents some challenges. Shown here are only some of the applications that HRM can be used for. Each has its own unique considerations, so the way the assays and even the way the analyses are performed can be a little different. This sometimes makes it difficult to figure out what's important for your experiments at first. The good news is the fundamentals don't change, and you already know how to adapt another related technique that's also very powerful with many different types of applications, PCR. Next slide. This slide shows a cartoon of how different homoduplexes and heteroduplexes can be formed when different DNA sequences are present in a sample. Here, three possible genotypes are under investigation: homozygous wild type, homozygous mutant, and heterozygote. Following amplification, four possible DNA duplexes can form. Two are homoduplexes, where there's a standard hydrogen bond binding the DNA base pairs. However, there are also two heteroduplexes, where there's a mismatch between the DNA bases. And this mismatch more strongly affects the melt profile. The mismatched bases melt more easily. This changes the shape of the melt and moves the curves to the left. Next slide. It's important to remember that HRM is a post-PCR analysis method. If your PCR doesn't work well, then whatever downstream method you're using is going to have problems. This is especially true with HRM because it's so tightly coupled to the PCR step and so sensitive. The good news is you probably already know a lot about PCR, and you can use that knowledge to advantage with a few good tips. The most attractive feature of HRM is speed. No post-PCR separation steps are required, like with HPLC or electrophoresis, mass spec, etc. HRM analysis of post-PCR melt profiles can provide a lot of information rapidly for multiple samples, especially when working with 96 or 3 to 4 well blocks. And when you're trying to fish out a rare variant among a background of other sequences, HRM is actually more sensitive than DNA sequencing. HRM can detect a variant down to about 2% of variant DNA in the background of normal DNA, while DNA sequencing can only detect down to 20% variant. So fundamentally, though, all we're doing is using temperature to melt the DNA and fluorescence to monitor the changes. And you've probably already been doing that with real-time PCR. Next slide. This isn't HRM yet. This is an example of a traditional melt curve analysis. But despite the name, we don't usually use the melt curve directly when checking our PCR products. What we're really using is the melt peak, which is the first derivative plot of the melt curve. The melt peak is a way to highlight and emphasize the differences in melt temperature, or TM. So the software is helping us to see the differences in TM that are there in the melt curve data but are hard to see otherwise. The melt peak helps us highlight the differences we want to focus on. So we actually do something very similar with HRM using a difference curve to visualize the differences in the melt curve shape that are important. There are a few differences between HRM and real-time PCR, though. One of them is the detection method. Next slide. So you can use both probes or intercalating dyes for HRM. Both approaches have their advantages and disadvantages. Labeled primers or probe-based detection methods offer great specificity and the potential, at least, to multiplex. However, they can be significantly more expensive than dye-based methods, and because of how they work, the probes must generally be designed for an individual template sequence, so it doesn't have the flexibility. In the next presentation, Dr. Winchell will present data from a real-world example where the relative advantages of both intercalating dyes and a labeled primer strategy were directly compared for a specific application. So I'm going to focus here on just the dyes, because these are the most flexible and affordable, and as a result of that, the intercalating dyes remain the most commonly used HRM strategy. So-called saturating dyes or third-generation dyes are generally used for HRM because cybergreen 1 is relatively toxic to amplification reactions. Consequently, cyber is used at low concentration in real-time PCR reactions. The dye doesn't intercalate at every possible position in the DNA sample. So when the DNA strands begin to melt, the dye can translocate and reintercalate back into the regions of the helix that remain double-stranded, reducing the resolution of the resulting melt curve. So the importance of this effect can vary greatly among different samples, but generally, third-generation dyes are a better option than cyber for most HRM applications. Third-generation dyes are much less toxic to amplification, so they can be used at higher concentrations. So when the strand melts, there's no room elsewhere in the molecule for the dye to relocate to another location. This makes the changing signal more definite and can provide clear melt plots and sequence differentiation. Next slide. Another little difference in HRM is the melt curve data are normalized to minimize the background differences among wells. This helps you look at differences due to sequence variation rather than PCR or sample quality differences. But normalization is not a cure-all. The reactions have to be pretty consistent. Next slide. From the normalized melt curve, we can create a difference plot, which is a way to emphasize and focus on the differences in melt curve shape. You can define the reference sequences, or the software will define the most abundant type as the reference. Then, at each temperature reading, the average fluorescence for the reference samples is subtracted from each individual sample, leaving us with a measure of difference from the reference. This process is repeated for each temperature point along the melt curve. The larger the difference in fluorescence readings at specific temperature, the larger the difference in the difference RFU graph. Large fluorescent differences between samples make analysis simpler. Next slide. The software will cluster similar curve shapes into groups. If you run known genotype controls, HRM software such as CFX Manager can auto-call samples to a genotype based on where they cluster due to curve shape. Next slide. HRM is largely an extended analysis of the melt curve, although it needs to be a good melt curve. HRM requires software to carry out the additional analyses, but HRM isn't just software. The optics and temperature control need to be sufficiently good. Not all real-time instruments can really do HRM, although these days there are several options that can. Each instrument has its own advantages and disadvantages, but we can't cover all those differences today. Next slide. So what level of resolution is required? The TM shifts for these SNPs can be very small. For class 4 SNPs, they can be less than 0.2 degrees, according to the literature. But these data do not factor in the size of the amplicon, the specificity of the primer pair, the dye being used, or a range of other factors. So what do these numbers really mean? Next slide. It turns out that class 4 SNP detection doesn't require 0.2 degree resolution. It's usually assumed that high-resolution melt requires readings across temperature range using very small increments. But more important than the ability to perform thin temperature slices is really the capability to detect small melt profile differences. Surprisingly, in the real world, the samples and the chemistry are typically the limiting factors. HRM just measures differences in melt characteristics. They can be because of different salt concentrations, different amounts of nucleic acid, inclusion of a small amount of dimers, or the sequence differences that we're actually trying to see. If your samples and procedures are not very uniform, the noise due to sample differences will overwhelm the signal you're interested in. Next slide. The take-home message is very simple. Small differences can be detected and distinguished. Here, using the CFX96 system, the most difficult to detect class 4 SNP can be resolved reliably. Next slide. Most of the assay design is similar to what's done for real-time PCR, but just as some primer sets that work fine for conventional PCR give too much noise for real-time PCR or don't have a high enough efficiency, some real-time PCR primer sets work fine for HRM, and others give too much background noise. So you're going to need to test each primer set, and you may need to redesign them, even if they work beautifully for real-time PCR. Next slide. Generally, shorter amplicons will be easier to discriminate as they will show larger signal differences and a better signal-to-noise ratio due to the lower overall background. None of these suggestions I'm giving today are hard and fast rules, though. You're going to see some nice data later today from a larger amplicon. There's absolutely no substitute for empirically testing your primer sets. If they don't give you the level of discrimination you want, optimize the conditions or redesign. Next slide. Here's an example of the effective amplicon size. Both primer sets work, but the smaller amplicon shows a better signal-to-noise ratio, which is going to help with some of the variation that creeps in when you start doing more samples and have a larger sample size. Next slide. In addition to amplicon size and location, good assay design, proper equipment, good samples, and consistent procedures are all required for robust HRM results. One aspect of proper assay design is well-optimized PCR reactions. Next slide. Even if your assay works well for real-time PCR already, there may be a small amount of non-specific amplification. In optimizing for real-time PCR, you may have even inadvertently selected for conditions that maximize the PCR efficiency at the expense of a small amount of non-specific amplification that doesn't really affect your real-time results noticeably, but will show up with the HRM analysis. It's therefore critical that the assays be very well optimized for HRM, and using a gradient thermal cycler is a really easy, helpful way to do this. So here are some data from some researchers in Singapore who were looking to develop a rapid screening tool to identify KRAS mutations in clinical cancer samples. They ran a gradient of annealing temperatures during the annealing step of the PCR, and the real-time reactions worked across a range of temperatures. When they did the HRM analysis downstream, they found that the increased background at some temperatures meant they couldn't properly cluster the samples. So the gradient allowed them to immediately identify the condition that was optimal for the HRM analysis. Next slide. I want to look at a few more aspects of assay design and focus on those which are important and those which are typically not. For this class 4 SNP, we're getting good discrimination with the very small temperature steps, as you might expect. Next slide. So even with the most difficult class 4 SNPs, temperature increments up to 0.5 degrees can work well, especially if you're trying to discriminate heterozygotes. So the point I'm trying to make here is that good assay design and consistency are the most valuable tools. Next slide. Other aspects of the experimental design can introduce problems, though. The volume differences shown here would only happen if you ran different reaction volumes in different runs. But I think it illustrates that even small volume differences can alter the melt profiles. So try to keep things the same among all of your samples. Next slide. Template input amount and template quality can be important for qPCR assay, but there's a really wide dynamic range for qPCR. For HRM, it's very important to keep those consistent. With the shorter amplicon, 3 different starting concentrations all fall pretty much together. But with the 100 base pair amplicon on the bottom here, the different genotypes still cluster, but you can see the curves separate into 3 different groups based on the starting amount. This noise is kind of okay in this small analysis, but imagine what would happen if you analyze 96 or 384 samples with different template amounts in each. The profiles, especially for the homozygotes, are going to sometimes overlap. The plateau effect and normalization that we do after PCR can help somewhat in equalizing these, but unintended variation is really the enemy of good HRM. Next slide. So, to summarize what I've been saying, you want to use only optimized reactions. The time spent optimizing up front always pays off in the end. Be consistent with reaction setup and reagent use. Results can vary depending on DNA template quality and the sequence of the amplicon. Avoid primer dimers and additional products that affect melting behavior, even if they don't show up in your real-time results. Use negative controls and standards where appropriate, and any ambiguous samples should be rerun or checked by another method. The other panelists are now going to be telling you a little bit about their work. Good luck with your experiments, and now back to you, John.
Adam, thanks for your presentation and for providing an overview of HRM. You've also set the tone for the talks to follow, so thank you very much. If you're just now joining our webinar, welcome. As I previously mentioned, we'll be conducting a live Q&A segment following the panelists' presentations. Again, please type your question for any or all of our panelists into the Ask a Question box on the lower left of your console, and then hit Submit. Our second panelist, Dr. Jonas Winchell, will now begin his presentation. Jonas?
Thank you, John. It's a pleasure to be with you today. The title of my talk is Applications of High-Resolution Melt Technology. My name is Jonas Winchell. I work for the Centers for Disease Control, and I'm a lab chief over a respiratory diseases branch lab. So much of this talk will be focused on pathogen detection and characterization. Next slide, please. This is a brief overview of how the talk will go. Briefly, the equipment where you'll see all the HRM data for this talk will be generated on the Rotogene 6000, which used to be owned by Corbett but is now owned by Qiagen. I will also talk a little bit about the sample preparation and primer design for this technology, go over briefly some real-time PCR chemistries that we've used in my lab, and then some of the applications on identifying SNPs for antibiotic resistance for some of the pathogens, also genotyping applications and speciation of a genus as well, and then we'll just have some follow-up conclusions. Next slide, please. The sample prep and target design, it is important to note for this technology that in our experiences we have seen differences depending on the procedure that you use to extract your nucleic acid does impact the results. So what you want to do is remain consistent between your experimentals and your controls. So if you've extracted your controls on a certain platform or a certain extraction kit, when you do eventually test your unknowns and experimentals that come in, you do want to do this exact same procedure when at all possible. If not, what happens is you get a little bit of variability, and it makes data interpretation a little bit more difficult. The direct testing of specimens is also dependent upon the particular chemistries you've used. We found a little bit of differences, whether we use intercalating dyes, which are very popular, and there's a lot of varieties of them out there, third-generation intercalating dyes or saturating dyes. But we've also used primers that are specific to the target that are labeled as well, and those are some chemistries that I'll go over. Also, we found that the template quantity in primary specimens does make a difference above the CT values of 37, 38. In that ballpark of a CT value, the HRM data become a little less reliable. So you do want to pay attention to the quantity that's in your specimen. Primer design is also dependent upon the application. We've used small amplicons, around 100 to 200 base pairs, all the way up to, I think we have the largest one out there in the published literature of around 1,900 base pair targets, and each have served their purposes. So you really have to just keep in mind what you're trying to accomplish with your design. Next slide, please. This is a specific chemistry from Invitrogen, which is called Lux, and that stands for light upon extension. In the left-hand schematic, you see there are 3 states of this labeled primer. So the primer is labeled actually on the 3' end with a FAM, and the 5' end actually has a complementary small piece tail hanging off of it, which will quench that in the unbound state, producing a hairpin structure with low fluorescence. And as it melts and then gets incorporated, the fluorescence increases as it gets extended into the product. So you have to realize that the amplicon itself is able to be melted with HRM technology because it does have fluorescence specifically incorporated, because this primer is designed against a specific target. On the right-hand side, it shows you just the amplification of it, but also it shows you a picture of the designer software. This is free from Invitrogen. I think it's actually even able to be downloaded from the internet, and there are different versions of this now. Next slide, please. Here are some characteristics of the Lux chemistry. As I said, the fluorophore is attached to the 3' end. It does contain a short sequence tail on the 5' that is complementary to the 3' end. This allows it to have a quenching effect by the terminal G's and C's on that tail. And the FAM label on the 3' end is usually off of the T or the C, about 2 or 3 bases in. And the software, the first version of the software only gave you the option of doing it off of a thymidine. The newer version has a T or a C choice. And it's important to keep in mind that the primer actually is extended and incorporated into the amplicon, which allows the melt to occur. Next slide, please. Here are some HRM curve applications with the Rotogene 6000. In our lab, we use both intercalating dyes and this Lux chemistry that I just described. Next slide, please. The first thing I'd like to show you is the detection of a single nucleotide polymorphism, SNP, which this is a particular case where in an organism known as Mycoplasma pneumoniae, which causes walking pneumonia, it can acquire resistance to a macrolides, class of antibiotics known as macrolides, by a single base change that's in the 23S rRNA gene. And in the upper kind of left-hand corner of this schematic on the right, at positions 2063 and 2064, there is a single mutation from an A to a C or G, which confers resistance. So what we had done, and historically they've just determined this through sequencing, but what we had done was to flank this region with primers and try to detect this one base mutation in real time using HRM. And because the shift goes from a 2 hydrogen bond to a 3 hydrogen bond, it's easy to detect with a melt, with a high resolution melt. Next slide, please. So to identify this SNP, we designed it to selectively amplify the 23S rRNA region where the mutations occurred. We used both Lux chemistry and Cyber Greener chemistry, which will and I'll show you the differences in a graph in a minute on their nuanced differences. Both of them were reliable to determine resistance, but I will show you a little bit different graphs in the different chemistries. And the Lux chemistry was actually better at determining resistance in clinical specimens. I should say specimens, not isolates. My apologies for that. And that's important because with isolates, either chemistry will work just fine because it's a nice pure culture and the targets are very clean and neat. In a mixed specimen, it's more, in our experience, it's more appropriate to use a specific labeled primer rather than an intercalating dye, presumably because the saturating dyes have a lot of higher backgrounds and there's some other amplicons that may confuse the interpretation of the data. So the Lux Chemistry worked better for us for actual clinical specimens. Next slide, please. This is the target region of the macrolide resistance. Along the top, we have the region of the 23S rRNA gene, where we have along the left-hand side different isolates of Pneumonia, the first three being the GenBank accession entry, and then we have two of the common types of sensitive strains. And about halfway down, you'll see the two underlined A's there, and those are the two positions that can change to a G or a C. And when we went through our freezers and looked at all the different isolates that we had, about halfway down that chart, you'll see the mutation at either position for 5 of these strains, which went from an A to a G, and that's what we detected in real time. Next slide, please. So this slide clearly shows the separation between the resistant and susceptible isolates. This is done with a cyber intercalating dye, and let me emphasize that this is a third-generation cyber. We do not attempt this with the cyber 1 dye as a first generation. So this is a third-generation intercalating dye, and it gives you really nice separation. Keep in mind, this is a single base pair mutation just from an A to a G, and we get really nice separation. Next slide, please. Same thing can be seen with the Lux Chemistry. In this case, the separation is less distinct, but still clear to be able to call resistant and susceptible isolate. And this was the more reliable chemistry when we did this with a primary specimen rather than just an isolate. Next slide, please. For HRM and genotyping, next slide, please. We did this originally for an organism known as Chlamydia acidicae, and this is a zoonotic respiratory pathogen. It causes severe pneumonia in people when they do acquire it. And we used Lux Chemistry for this. We designed primers to amplify a 274 base pair region, and from this one amplicon, we were able to discriminate 5 of the 6 different genotypes of C. psittaceae. Next slide, please. This shows you the genetic region we targeted. It's an outer membrane protein product from this gene. Along the left-hand columns, you see the genotypes A through F in a consensus sequence. And then at the top portion, the 5' end, we have the underlined portion, which is our forward primer. That's a Lux labeled primer. And all the way down at the bottom there, we have where the reverse primer would bind as well. And so within this amplicon region, you can see all the different bases that are different between the different genotypes. And when we amplify this up, we do have nice amplification followed by the HRM, which could take apart A through F. Next slide, please. And what we find here is going from right to left, we have genotypes E, A, B, D, and F, and C. So D and F were so similar that we could not tease them apart with HRM technology. We just designed another specific TACMAN chemistry to amplify F in order to achieve the goal of getting all of these apart. But you can see that they're nicely spread apart and easily determined that you can genotype these routinely and reliably. Next slide, please. We also use this technology, a little bit different approach. This is for the genus of Brucella, which, if any of you are familiar with this, know that they're very, very similar species. They're all kind of shades of gray, as it were. So we targeted different loci for each of these species, and we did. This is with an intercalating dye, and we did all different melt curves with this. In panel A, you can see how melitensis is separated out away from all the other species. In panel B, same thing, except it's B canis. In C, we take away all the marine strains versus all the other species. And D, we take away the only known B neotome that's out there. And E, we can separate both obus and abortus away from all other species. And then F, we have a real-time just plus-minus amplification detection of B suis. And this has important implications in bioterrorism field, also the animal industry as well. So this was just published back in January for the HRM technology for this genus. Next slide, please. We also genotyped M. pneumonia. As I said before, this is a common cause of walking pneumonia. It's a community-acquired pneumonia. And historically, it's been separated into type 1 and type 2 categories. This was done through a nested PCR and a really laborious RFLP analysis, and it's very time-consuming and messy. So what we tried to do was to see if we could do this using real-time PCR followed by HRM. Again, keeping in mind this is a closed system. There are no manipulations after the initial amplification. It follows straight into an HRM. We used cyber green chemistry. Again, this is the third-generation cyber dyes. We targeted, by necessity, actually, a 1900 base pair amplicon because within this organism there is a large amount of repetitive units. And so we had to actually flank a pretty large unit in order to get specific primers that would only bind once on each of the sides of this amplicon. And this is designed to flank a P1 gene, which is known to be the target to group these into a 1 or 2. And despite the large size, the differentiation was clearly evident. Next slide, please. You can see the very large separation of the type 1 isolates versus type 2. The good thing about the rotogene technology as well, next slide please, is the fact that you can represent this data in different ways. This is a difference mode or difference graph, and basically what you do is you just tell the computer to normalize everything. In this case, we put the type 2 isolates, we normalize that to 0, and anything that deviates significantly from those, it shows you that nice big curve for the type 1 isolates. So either way, it's easy to see a type 1 versus a type 2 with this technology. Next slide, please. It also afforded us the opportunity when we went through our repository of strains that the powerful nature of this technology, we were able to identify variants of each of these two types. It's been documented in the literature that they do exist, and we actually found a few. All the way to the right, we have a prototypical type 1, and all the way to the left, we have a prototypical type 2. And then we have 3 different curves that melt at different formations throughout the temperature range here. And then isolate 549 and isolate 684, those are type 1 variants. And then isolate 3 technically is a type 2 variant, but when we went back and analyzed it, next slide please, you'll see that we found very unique regions. This is the gene or the amplicon portion that we amplified, and along the top we have a type 2. The second one down is a type 1 strain, and then finally is this isolate 3. And when you're looking at these different colors, what we found, which is very interesting finding, is that when you find the pink sequences, those are identical to a type 2. The aquamarine blue are identical to type 1, and then the yellow ones that we found were unique to isolate 3. So the point being here that the HRM technology allowed us to, in real time, very rapidly, able to follow up on a very weird isolate from a clinical setting, in which this hybrid of a pathogen allowed us to do further characterization of the sequencing, and we could do that very rapidly because of this technology. Next slide, please. So our conclusions are that it is an extremely flexible and powerful tool, allows you to quickly identify genetic differences. We were able to determine antibiotic resistance in some cases, genotypes, speciation, SNP detection. We have used this in different chemistries used in this technology. We have had success with both the intercalating dyes and the Lux technology. And the Lux seemed to be a little bit more appropriate for testing primary specimens due to the background fluorescence. And I can't stress enough on how much this field has advanced in the last couple of years, just based upon the better master mixes and the better and more exquisitely sensitive and calibrated machines and platforms that are out there. And it is just coming hugely for our purposes here at CDC. And with that, I'd like to next slide, please, just like to acknowledge some of the people who have are colleagues of mine at CDC or collaborators at the University of Georgia. And with that concludes the talk. Back to you, John.
Thank you, Jonas. And that was a great description of the equipment, sample prep and primer design procedures, and the real-time chemistries involved in working with HRM. And I really think your discussion of HRM for genotyping and for identifying SNPs that are associated with antibiotic resistance clearly demonstrates the specific capabilities of this technique. So thanks so much. Before we go on, let me remind everyone of our live Q&A segment that comes right after the panelists have made all their presentations. Please type your questions into the Ask a Question box on the lower left of your console and then hit Submit. Our third panelist, Dr. Kim Delanier, is ready to examine the applications of HRM in a diagnostic setting. Kim?
So thank you for this introduction, John. In the next 20 minutes, I will briefly point out the setup used in our lab for BRCA1 and 2 mutation detection with high-resolution melting. Next slide, please. The two main applications for which HRM is used in our lab are first the mutation scanning, which is used to quickly eliminate wild-type sequences, and second, genotyping with the help of unlabeled probes, which can be used to genotype clinically important SNPs, but also as a supplement to mutation scanning to reduce the sequencing burden afterwards. The rest of this talk will mainly focus on the mutation scanning application and the genotyping as a supplement to mutation scanning. Next slide, please. All the results presented here today can be found in these two articles. The first one describes the mutation scanning setup in detail on two different HRM platforms, and the second one, which was published last year, provides a protocol for the unlabeled probe genotyping. Next slide, please. We chose BRCA1 and BRCA2 as candidate genes to evaluate our HRM applications. After evaluating HRM assays for these two genes, assays for other disorders could easily be optimized. So first of all, why exactly did we choose BRCA1 and 2? These two genes are a real challenge for mutation scanning. They are both large multi-exon genes. BRCA1 has 22 coding exons and has a remarkable feature, a large exon 11, which represents about 50% of the coding sequence. BRCA2 is an even larger gene with 26 coding exons and two large exons 10 and 11, and these two exons represent about 60% of the coding sequence. Next slide, please. Furthermore, there are no mutation hotspots, and those two genes are very polymorphic. Until May 2007, our mutation scanning strategy consisted of direct sequencing of both large exons 11 and DGE or denaturing gradient gel electrophoresis for all other coding exons. So to reduce the costs and the workload and to be able to screen very large groups of patients in a research setting, an HRM setup was designed. Next slide, please. We were looking for an easy setup, but since there are no mutation hotspots present, we needed to cover the complete coding region and splice sites of both BRCA1 and 2. The only clue we had back then was that the maximum fragment length could only be 400 base pairs to guarantee a maximum sensitivity. This was published by Reed and colleagues. So our first step was actually the primer design. We designed primers by the following rules. So first, we checked the absence of SNPs in the primer binding sites with the ensemble genome sequence database. The annealing temperatures were all chosen around 52 degrees, which might seem low, but BRCA1 and 2 are very AT-rich genes. Other specific features of the primers, like melting temperature, hairpin formation, were checked with a freely available online tool. The specificity of the primers was controlled with UCSC in silico PCR program. And last, to simplify the sequencing reactions afterwards, universal and 13 tails were fused with the primers. Next slide, please. Our primer design resulted in 44 amplicons for BRCA1 and 68 amplicons for BRCA2. So in total, we have 112 PCR amplicons, and they can all be amplified with the same touchdown PCR program, allowing us a semi-automated approach. Two examples are shown here, and as you can see, the PCR products are strong and specific, which are the basic requirements to obtain a good melting profile with HRM. Next slide, please. This slide shows some results from our mutation scanning on the light scanner instrument. The first panel shows BRCA2 exon 2. The melting curves and the different plots are shown. The red curves are all samples with a frequent intronic polymorphism. The blue curve represents another sequence variant, and the green curve covers both peaks and has both sequence variants. The panel on the right shows two founder mutations in our Belgian population, and as you can see, they can both be easily detected with our HRM amplicons. Next slide, please. So next, some results on the light cycler 480 from Roche. Again, BRCA2 exon 5-6 is given, but on this instrument we had to increase the standard sensitivity setting from 0.3 to 0.5 to detect the same splice site mutation as mentioned on the previous slide. For a few of our positive controls tested, we even had to increase the settings to 0.7 to be able to detect all variants. Next slide, please. Then some results on the CFX384. The two examples are shown here, with again in the upper panels the melting curves and below the normalized difference plots. And as you can see, the deletions were all easily distinguished from the wild type samples. Next slide, please. So in overview, for the sensitivity and the specificity of the three evaluated instruments, on the light scanner we were able to detect all positive controls tested with a standard sensitivity setting. In a blind screening of 22 patients, we detected 32 false positives, leading to a specificity of almost 99%. On the light cycler 480, we could only detect all positive controls when we increased the sensitivity setting to 0.7, which led to a small reduce in specificity compared to the light scanner instrument. By analyzing the curves on the 0.3 sensitivity setting, we missed 87 mutations, obtaining a specificity comparable with the light scanner instrument, but of course missing important positive control samples. Then on the CFX384 instrument, all variants were easily detected with the standard sensitivity setting, and we found a specificity from around 99%, but this is only an estimation compared to the other numbers, since we evaluated 30 positive controls and only 500 amplicons to determine the specificity. Next slide, please. Then I would like to point out some possible pitfalls in HRM mutation scanning. So first, the detection of homozygous SNPs. Those variants are rather detected by a shift in melting temperature, as shown in a difference plot below. And as you can see, the blue curve is shifted to the left compared to the heterozygous samples. In a study published by Liu and colleagues, they concluded that 4% of all SNPs present in the human genome will remain undetectable with HRM. So as a solution, you can mix your sample with wild-type DNA. We actually tested this in our lab and concluded that about adding 10% of wild-type DNA is enough to be able to detect homozygous SNPs. Furthermore, this can also be solved by designing an unlabeled probe assay for these SNPs, but I will come back to this later. Next slide, please. A second possible pitfall is that different variants may cluster together in one group. The examples are shown on the slide. On your left, 3 different aberrations are grouped together, As I said a few slides ago, the aim of HRM mutation scanning is to quickly eliminate your wild-type sequences. So in theory, it should only be used as a pre-screening technique. If you have unknown variants, you can easily solve this by sequencing all samples with an aberrant difference curve. On the right, another example is shown where different SNPs are grouped together because they are only 5 base pairs away from each other and hence generate similar melting profiles. But since these are known SNPs, this can easily be solved by including an unlabeled probe in your reaction. Next slide, please. The last pitfall I wanted to show is the detection of variants that are present in homopolymeric repeats, like this variant shown on the slide. This is an insert of a T in a stretch of 12 Ts, and as you can see, both instruments have difficulties detecting this variant because of the very small melting temperature difference generated. Next slide, please. So to continue the story of our easy setup, after covering the complete coding region of BRCA1 and 2, we still had the problem that BRCA1 and 2 are very polymorphic genes. So a lot of our samples still had to be sequenced after performing our mutation scanning analysis. So we designed 14 unlabeled probe assays for the 14 most frequent SNPs in BRCA1 and 2, and 2 of those probes contained an additional mismatch for another SNP a few base pairs upstream. To not further complicate our setup, we wanted to use the same primers as in our mutation scanning assays. We designed our probes with the light scanner primer design software. The melting temperature of the probes was about 60 degrees. The maximum length of the probes was about 35 base pairs, and we designed a few probes when cross-complementarity with one of the primers was found. Next slide, please. For the optimization of these assays, we used 15 control samples, 5 wild-type, 5 heterozygous, and 5 homozygous samples. Important parameters to optimize seem to be the magnesium chloride concentration and the number of PCR cycles needed. For some of our assays, we even needed to increase this number to 60 to obtain a good genotyping profile. The sensitivity and the specificity were evaluated with a blind screening of 95 samples, and all samples were correctly genotyped. Next slide, please. So this is an example of our setup now, shown for BRCA2 exon 2. We first performed the mutation scanning part, as shown on the left. With this analysis, you can see there are 2 samples who show an aberrant melting curve, so normally these 2 samples would need to be sequenced. But in parallel, the same samples are genotyped with an unlabeled probe assay, covering a frequent SNP in this amplicon. This shows us the presence of the SNP in heterozygous form in the 2 aberrant red melting curves, and in this way, no further sequencing is needed. Next slide, please. Another example why these probes are useful is shown in this slide. In the mutation scanning panel, 3 groups can be easily distinguished. Unfortunately, different variants are grouped together. The wild-type sequences are grouped together with the homozygous mutant SNPs, and since this amplicon has 2 A to G SNPs only a few base pairs away from each other, the software isn't able to separate those from each other either. So when we look at the genotyping assay with the unlabeled probe that actually covers both SNPs, all 5 groups present in this sample set can be easily distinguished. Next slide, please. So in conclusion, we designed an HRM setup to reduce the cost and the workload, and we can say now we reached our goal. A screening of 11 patients with HRM takes about 3 days, compared to 5 or 6 weeks with DGGE and direct sequencing like it was before. Next slide, please. Concerning the costs, we had another large drop in sequencing reactions after supplementing our assays with the probe assays. Sequencing reactions are normally the major cost, so if you follow in the graph, you can see the drop was tremendously. So in an overall conclusion, HRM is a cost-efficient, sensitive, a specific and a high-throughput technique, and it's simple enough to be readily implemented in a diagnostic setting. Next slide, please. Last but not least, I would like to thank all these people that contributed to these studies, and of course you for your attention. Thank you. Back to you, John.
And thank you, Kim, for zeroing in on the use of HRM in BRCA1 and 2 mutation scanning. Boy, you've certainly made the case that this approach is cost-efficient, sensitive, specific, high throughput, and simple, so thank you. I want to congratulate all the panelists for their enlightening discussions of HRM. In addition to describing some potentially novel applications for those already using the technique, I think you've made some converts in our audience, certainly by the number of questions we're getting, who have been thinking about incorporating HRM into their repertoire of the research tools. As I said, people have been sending in questions. We're going to begin the Q&A in a sec, so let me take a look at the first question, which is for Adam. Adam, what about the detection of class 4 SNPs in larger fragments than 50 base pairs? I assume PCR fragments for HRM routinely are somewhere between 200 to 400 base pairs.
Yeah, so I think I actually showed a little bit of data for a somewhat larger amplicon. I would say commonly, just because of the transition from qPCR, people start off working with 200 to 400 base pair amplicons. Jonas showed some data with what even for qPCR is a very large amplicon, I think 1,900 bases. I've done over 1,000 base pairs myself. Generally speaking, the larger ones, you're going to have a lot of difficulty trying to get a class 4 SNP. Usually 200 to 400 base pairs, you can often get class 4 SNPs to resolve with those. But it's not just, I don't want to give the impression with my presentation that it's just size. Melt domains matter as well. Sometimes a 200 base pair fragment is going to work beautifully. Other times, a 300 base pair fragment might actually work better. Usually, though, the smaller you get, the better. And I would actually recommend that you try and push things down to small amplicon size when you can for the information you're looking for. The one other caveat I would have is, again, it matters a lot depending on if you're looking for a single class 4 SNP, you're probably going to need a much tighter resolution. You're probably going to do better with a smaller amplicon. Some things that I've done with HRM involve looking at things like indels. And if you're looking at an indel, you can easily do that with a much, much larger amplicon.
Adam, we have another question. Is a high-fidelity thermostable DNA polymerase required?
No, it's not. In the early days, people used their own homebrew PCR mixes. There are still some people out there doing HRM that way. I would say, again, I work for a company, so take this with a grain of salt, if you will. But generally, commercial HRM mixes are going to work better for you. So I would just go with the polymerase that's in there. But if you have a fidelity error early on that gets amplified, that's going to throw your HRM out analysis off. But most of the time, those errors are going to accumulate in the later rounds where they're not going to show up as much. And usually you're actually better off using an enzyme that isn't necessarily super high fidelity, but that works well and that's matched well with the dye that you're using. And because of low cost, you can actually run the same sample twice. So if you do get an anomalous result, rerun it.
Adam, another question. Do you need to have a positive control template for each of the variants in every batch of HRM?
No, you don't. It depends on what you're, again, I hate to give this completely useless answer that it totally depends on what you're doing, but it does. If you want to actually call genotypes, then you have to have a control sample that the software can use to actually call the genotypes for you. Different softwares work differently. Ours has the ability to sort of combine multiple runs together. Some others do not. But generally speaking, you don't need to have positive controls at all. But if you're trying to call genotypes, they're very, very helpful. Without them, you're kind of binning. And what you're doing, if you don't have the controls at all, is you're just binning things into this is one group, that's another group, this is a third group, and then you can always go back and assign what those sort of bins are later. So I would say that they aren't required, but they definitely help your analysis. And if you want to get the most out of the software tools, then they're really helpful for that.
Okay, Adam, the next question is directed to you and also to Jonas, so we'll start with you, Adam, since you're on a roll. In addition to discriminating between variant types, can you quantify?
I would say yes, but there's better ways of doing it. There's at least theoretical ways of quantifying. I assume what the question's really getting at is looking at your melt profile, which might have, say, a mixture of things in it, and trying to quantify what percentage in that particular well is variant A and what percentage is variant B. And I would say that there are better ways of getting at that level of quantification. You can get sort of semi-quantitative numbers, certainly. You actually can get quantitative numbers out, but it's tricky. So I think there's better ways of doing it. Jonas?
Yeah, I'd agree with Adam. I was just going to interpret that a little bit differently with on a spin on the lower limit of detection for a reliable melt curve. That in our experience, we've gotten down to between 50 and 100 femtograms, if that's in the starting input of template. But as Adam said, if you want to purely quantitate, there are different ways to do that probably.
Okay, Jonas, I have a question for you now. What fully saturating dsDNA binding dye is the best for HRM analysis? Pros and cons, any unbiased manufacturer-free evidence?
Yeah, well, I guess the best answer to that is there is no one best dye. It really is application dependent. It's amplicon size dependent. It's your primer designs, and it's also platform dependent. So in our experience, we've tried, I don't know, 3 or 4 or 5 different types of intercalating dyes, and we've found that one manufacturer might work for a certain application, whereas another one will work for a totally different application. And I think the thing that I guess the folks can count on is that the companies out there are continually modifying and kind of improving all the different dyes. And so you have to continually scan the market to see, what's going to be the newest thing to possibly suit your needs. And the one thing that we did notice with some of the manufacturers with some of our sets is that the problem that we would run into with certain sets was NTC problems. The primer dimers would come up in our no template control, whereas if we switched out to a different one, we would be fine with that, and then we may have that exact same problem, but a totally different master mix with a different primer set. So there is no real clear-cut answer as a best dye. It's really something where you have to empirically test. It may serve you well to scan the literature and see what you're trying to achieve and see what other people have done, and then the one that fits your application the best, you might want to order those kind of reagents and try those first.
Jonas, someone from the audience is wondering why you aren't looking at difference plots.
Oh, we do. It's just, I think it comes down to personal preference. We look at both the difference mode graph and the normalized kind of line curves, I guess. And we've published it both ways. It's just a personal preference. I don't know if any of the other people on the line have an opinion on that, but we've used both, so it's not that I don't look at them.
And Jonas, when multiple genotypes are shown on a slide, were they from a single HRM reaction or multiple reactions and compiled in one graph for seminar purposes?
No, those are actual. Well, I mean, this is all from the Rotogene Q, Rotogene 6000. Okay, so on that, what we would do is select the well numbers to align on that graph, but within any one tube, that's a single HRM reaction. But to obviously compare sample 1, sample 2, and sample 3, etc., those are all just clicked on or off to show where they're melting. So there's not a competition within the tube itself, and it wasn't, I guess, modified for a presentation, that is exactly, just it's based upon well selection as how you want to show your data.
Jonas, is it possible or easy to do multiplex analysis if the melts are more than one target in the same tube with HRM?
Well, there's a couple different ways to answer that. One is the fact that you're getting multiple melts on a single amplicon is essentially can be interpreted as a multiplex. But in the true sense of a multiplex with different dye channels, I believe my familiarity is with the Rotogene Q, and I think that's a single channel on the green channel that we melt with an HRM. The way that we have multiplexed before, which I didn't show you in this presentation, but we've done some experiments with the TB folks, and what we've been able to do is to mix chemistries, and meaning we can take a intercalating dye master mix and use unlabeled primers and flank a region, and then look at those melt curves. But also within that exact same master mix, we can add a TACMAN probe, which specifically covers a point mutation. It's a locked nucleic acid probe, and we can put a different floor on that. And so we can look in the green channel for the intercalating dye and see the melts and interpret our data that way. And then we can click on a totally separate channel to see if that has also provided a signal, and then we can say, well, it specifically has this mutation as well. So you can mix chemistries to a certain extent to show, for that study, that was for a multi-drug resistant TB detection, which is currently accepted and being published in JCM, which will show you how to mix chemistries, I guess. I don't know if that answered, hopefully that answered the question.
Okay. Hey, Kim, we didn't forget about you. Actually have two questions that are very similar, so I'm going to ask them together. If we have more than one SNP in the amplification product, can HRM effectively be used to genotype samples? Now, hold on a second while I go to the second similar question. Okay, hold on. It's down at the bottom. We have a lot of, here we go. All right, so the other question is, can you really genotype based on the melting profile? Isn't it possible that a different SNP gives you the exact same profile?
Well, this is a very interesting question, and it actually depends on the SNPs itself and or the amplicons. So there were a few examples in my presentation, and for some SNPs that are close together and generate a similar melting difference, it isn't possible to really genotype both SNPs in one amplicon. But of course, there are a lot of examples of SNPs that are a few base pairs away from each other that actually do generate a totally different spot or melting curve. So it's very dependent on your sequence environment and the SNPs itself. If you have two G to A transversions, it's a totally different story than a G to a C or a to a T.
Kim, what 3' block is preferred for HRM unlabeled probes?
We actually ligate a C3 spacer at the 3' end, but those are quite expensive actually. So for the optimization, you can actually try or optimize your assays with only some mismatches at the 3' end. And when you actually have found the right assay, you can order one with a C3 spacer, because it just works with the mismatches for a few times, but then actually you get some amplification. So the probe serves as a primer, and then your assay goes backwards.
Next question actually is based on your previous answer there. What is additional mismatch?
Yeah, those additional mismatches, I think it wasn't really clear in the presentation, but I actually meant, so we had two probes with an additional mismatch, and that's actually a SNP, a few base pairs upstream of the other SNP, where we included an additional mismatch in the probe. So we can actually genotype both SNPs at once in one assay.
And a question for Adam. What is the mechanism by which an unlabeled probe increases HRM resolution?
There's actually a couple ways that probes can increase HRM resolution. I'm assuming that this is using a dye-based method, and really what you're doing is it's a way to kind of cheat the system and go after a little bit larger amplicon, but then have the melt really focused on just a small portion of that. And so just like I was suggesting that not always, but often it helps to get down to a really small amplicon. If you've got an unlabeled probe in there, but with some dye, you can actually see the melting of that probe coming off, and the thermodynamics of how that melt and is affected by a single base pair difference is going to be much bigger than how the entire PCR amplicon is going to melt.
Well, thank you for the answer. Well, it looks like we've run out of time, but please note that the webinar will be archived for 6 months on our website, www.genegnews.com. If you missed parts of it, you can watch it again, or you can recommend it to your colleagues and friends. In addition, some of the questions are going to be posted on our new section of our website called Gen Connect. It's our new social networking platform. You can see it by going to www.genegnews.com/Gen Connect. Thanks again to our panel for their outstanding presentations. I also want to say thank you to our audience for your attention and for your very thoughtful questions about various topics brought up during the webinar. And thank you to our sponsor, Bio-Rad Laboratories, who made this event possible. Shortly, we'll be sending you a survey, and we would appreciate it if you would provide us feedback on this webinar. Please look out for it and kindly give us your thoughts, as this will help us continue to bring you topical and timely webinars in the future. Thank you very much.